I'm going to talk about computational MR and AI for understanding the fetal brain development, but also with a little bit of outlook on infant development, because I think the two create a continuum and we should not just look at them in isolation. First of all, I need some explanation to do why to put AI in this. I know it's very popular these days, but I still thought that it's important to talk how this can help our work as scientists. So I'm not going to talk about clinical uses or anything like that, but to look at it as a tool. And also, I think maybe it's a little bit like computational image processing is now replaced by AI as a label, but nevertheless, the two are kind of has always been related. So it's not entirely new. And you will see in my talk what I mean by that. But first of all, let me just tell you why I think studying the brain development is fascinating or interesting. You know, it's a very it's trivial, I think, that the brain undergoes rapid development and it's a complicated process, but many people don't realize some of the facts that I have brought. It's more for a popular lecture. I know most of you have heard of it, but I still think it's fascinating how the brain gets from this neural crest to a functioning, crying baby by birth. So I just brought these facts that during gestation in humans, 250,000 neurons are born every minute and 1 million synapses are made every second. That these are kind of estimates from scientific studies. And now that we talk a lot about AI, if we count how many synapses a brain has, it's 600 to 1,000 trillion. So it's still 1,000 times larger than the parameters in a GPT model. So we are still not there yet. And someone estimated the storage capacity. I don't exactly know how, but they came up with this, that the brain has 2,500 terabyte storage. There you go. But most importantly, at birth, I would say that we have almost everything when it comes to the major anatomical pathways. They're still in development, but at least we see them. And we also know that sensory learning and picking up language and it already begins in utero. There are studies for that. And the other thing why I am fascinated about brain development recently is that it's the only known biological model or only known phenomenon in nature, which is how an intelligent system develops basically from nothing or from a bunch of cells to an intelligent and also conscious brain. So I think we should not underestimate the value of studying brain development. Now, you see that this is a very complex process, and any disease or any genetic environmental factors influencing it can have tremendous consequences. So looking at the, let me just close this. Looking at the clinical uses, I'm sorry, I just had a pop-up window. Sorry. Oh, where am I? Because I know. Okay, good. So diseases affecting brain development are actually not that rare. So according to recent statistics, premature births can be up to 10%. Most of them are mild and not very low rate preemies, but still it's not that uncommon. Congenital heart defects also affect brain development and they happen up to 1% of birth, and also genetic abnormalities, syndromes of different reasons are also 1 to 3%. Now, what are the clinical characteristics and what can MRI help us to learn about these conditions? So, for example, in prematurity, we know that the development of brain connections are impaired. This could be caused by, or there's mechanisms that there's a dysfunction of the pre-myelinating oligodendrocytes. There's white matter injury that one can see on the MR or even ultrasound images. There's also bleeding of different kinds if it's a very premature infant. And what we know from longer term studies is that they do face impaired long-term cognitive problems, especially in the domain of executive functions, again, as a function of how early they were born. Congenital heart defects have a similar effect. They show white matter injury with a slightly different pattern. Smaller brain volumes, even in utero, have been found. And as these children now are getting into adult age because of the improvement of the surgical techniques, they survive for a long time. We see that they do have sometimes academic troubles. Executive function deficits are also quite common. And genetic abnormalities of other kinds it's a very heterogeneous group, but I just highlight spina bifida because this is one of the conditions that we study in our research group because of local clinical reasons. So today I'm going to talk about 4 topics. You could, or I could have used computational imaging as a replacement for AI, but I'm going to focus more on the machine learning for the developing brain segmentation. Then I'm going to look at connectivity and analysis. And then a new topic, which we do with a collaborative partner, is developmental brain age estimation from MR images. And then in the second part of my talk, I'm going to present you some practical uses of these approaches, latest findings in normal development, as well as in the pathological conditions that I was mentioning. Now, I'm grateful for Maria for mentioning challenges and what we can do with the data. I don't have to detail that. We face challenges in the fetal brain because of motion. What is a similar challenge is that we also have pathological brains, as you see in the middle. Most of the fetal MR data that we have are pathological. So while it's great to have these large-scale normal databases, we still need to have methods that work on pathological brains. In the neonatal setting, you see they are imaged in natural sleep, which has also some limitations. They sometimes wake up and you need special scanning settings for that. Now, moving to image segmentation, what is actually image segmentation? So in general, segmentation is the delineation of a structure on an image. What I would say that it's something that a kindergarten child can do, cutting out something out of an image, because for the human brain, it's a pretty easy task to do. You immediately realize where the horse is because of this learned semantic concepts. But for a computer, this is a big problem, and that has been the focus of much research even before the AI craziness took off. People were developing these methods. Now, we wanted to move this field forward. So as a first step in 2020 and published 21, we created an open data set, which is called the Fetal data set, which contained a mix of normal but also pathological anonymous cases, which were hand segmented for 7 different anatomical structures. This is not as good quality as the DHCP, but it was actually, the first of its kind. And how we can use this data, we can use it, for example, to benchmark and develop new image segmentation algorithms. And once we have segmentations, we can do quantification of brain growth, as we have seen in the previous talk already. Now, this challenge that we created has gone through different iterations. I just brought a summary, also resulted in a lot of very good papers, which I'm grateful for. Now, first we started with a smaller data set, and then we started to introduce multicentric data. So from 2022, we already pulled data from UCSF, from Vienna, from Lausanne as a hidden test data set. And in 24, Mikhail, we introduced a completely new challenge, how to get fetal biometry directly from images. So we had to do this almost like the obstetrics biometry data manually annotated. Now, I'm not going to go into a lot of details, but what we noticed from the beginning is that people tended to use UNet and NUNet, which were mostly working out of the box. And we did not see a lot of difference. So you see the different groups, NVIDIA being the top in this challenge, their own research team, they perform similarly. So there's a plateau. We could not really improve segmentation accuracy beyond a certain point. Maybe the human annotators were not good enough, or maybe the data was just not good enough. But what we also concluded in the multicentric challenge is that there is a need for domain agnostic segmentation approaches. And at that time, there was no SimSeg available, at least in the beginning, there was not yet. You see from this picture how different can fetal images be when you pull them from different hospitals or different field strength. You have the low field from King's in the test data, or if you, for example, use T1 instead of T2. So the segmentation approaches are not domain agnostic by default. We need to implement some tricks for these approaches to be agnostic to the image domain. So SimFAC came at the right time. This is not from us. This is from the Boston group. The theory is that, which is a very surprising one, that you don't actually need realistic images. What you need is a very large amount of randomly synthetically generated images, which you generate by taking in a template, which is almost like a label map of an atlas, and you randomly shuffle the image intensity values on a large scale during training time. And then by doing this, basically your segmentation network becomes agnostic to the underlying contrast. The problem with it, what we notice for the fetal brain, is that it doesn't work on pathologies, and there is no real way to augment your network on a large scale for pathologies. So what one of my postdocs was working on is we wanted to have a generator that not only generates the image intensity perturbations, but also generates pathology perturbations on an image. So you take a normal fetal image, and then in the label map space, we introduce different variations that we think are associated with pathologies. We can learn that, but in this paper, it's done by very simple means. For example, we can use the DHCP and basically create artificially ventriculomegaly cases. So this is how these data looks like. Basically, we generate as many of these as training epochs and training runs, so a lot of times. Now, if we look at cases like that, this is a spina bifida fetus. It's very difficult to use any network trained on normal data. Of course, you can say, why don't you just annotate pathological cases? First, it's very time consuming. You don't always have a very nice age distribution of pathological cases, and not everyone has pathological cases or access to pathological cases. So what we did is we tried a different combination of training data. It's in this paper, I'm not gonna detail, to make sure that they represent normal as well as pathological development. What came out of this paper is that it's still very challenging, but one of the experiments had a very high Dye score for the ventricles, of course, because they were trained on a large amount of artificially dilated ventricles. So we were a little bit disappointed, and we thought we maybe give a try and investigate another approach. So we moved a little bit towards synthesizing these images with learning. Right now, we're not yet there, but I can introduce this topic like this. So we started with a network like this, which is the SPADE-GAN. It's a pix-to-pix based network that takes as input a semantic label map that you see on the left, and it generates a photorealistic image like you see on the right. This is already quite old. That's a GAN approach. We wanted to use a little bit later approaches like the stable diffusion. So what we did is we conditioned the stable diffusion network on the semantic label inputs, and we wanted to keep the semantic label inputs as simple as possible. So we just had 3 labels: cortex, ventricles, so fluid spaces, and everything else, because we did not want to over-specify, because then it will be dependent on the segmentation approach too much. And what the network learned is how to synthesize a realistic-looking MR image out of these label inputs with all the basal ganglia or the MR artifacts. Even the plexus was kind of learned by this network, how to put it. Now, what this allows us to do is basically have a simulation of different pathologies by simple operations and kind of a synthetic MR image that looks real. I mean, it's not a real patient, but at least we thought it's useful for training a segmentation network. So the purpose of all of this is really downstream applications and not really to just synthesize images for entertainment because they're not so good. What we developed and described in this paper, hopefully published soon, is different simple filters that simulate pathologies. For example, asymmetric ventriculomegaly. So we could tell if the left or the right ventricle should be big and by how much. We simulated cerebellar and pontocerebellar hypoplasia, again, by very simple ways. And my postdoc, Misha, also combined different training data, like healthy subjects, simulated real simulated images from healthy subjects, and so on and so forth, to come up with a final network that is able to segment fetuses with spina bifida and other conditions with relatively good accuracy. Moving on, I just want to highlight it. So this is, we take in a real image with a real label map and basically generate derivatives of these input image, which relate to different kind of pathologies like cerebellar hypoplasia, for example. So you see that the ventricles were pretty challenging to segment. Now that was structural MR imaging, but as you know, MR can do much more than that. For connectivity quantification, I'm just highlighting the methods we developed or implemented for brain connectomics research. But before I do that, just a quick recap about what brain connectivity is. Usually in the MR community, people talk about structural connectivity and functional. Structural meaning how strongly region A and B are connected by actual physical axonal bundles or the reconstruction of them based on MR image. And you do that with DTI or in the anatomy world with invasive tracers or diffusing tracers. Now, bold fMRI allows us to quantify functional connectivity, which is more like a statistical concept, covariation or correlate of time courses you extract from two regions. And there's a connection if these two regions are correlated with each other. That's to put it very simple. Now, when you work with structural connectomic data, usually what you have is large connectivity networks. And there has been a lot of studies that were trying to make order in how we interpret these findings. And we like this very much from Martin van den Heuvel and Olaf Sporns. It's a concept that they propose that the brain is basically operating in a space determined by optimized integration and optimized segregation. So this is a duality of how information is processed in the brain. And what they proposed is this blue line, which is the optimum between integration and segregation for healthy individuals. And they propose that diseases are basically the transitions or moving away from this optimal zone, and that creates a morphospace to interpret any diseases. So we thought with Anna from my group during her PhD, why don't we look at how different neonatal conditions and development itself is located on this artificial morphospace that van den Heuvel and colleagues proposed. So what we did is we came up with a normalized metric for integration and segregation based on some hypothesis randomized networks. So these are kind of hopefully comparable across the different groups. And we took developing normal controls, congenital heart defect infants, prematurely born infants, and spina bifida infants. And this is how it looks. So we see this kind of similar orientation and that there's a great overlap, but also some conditions do occupy a slightly different territory in this morphospace. Now, so far so good, not extremely useful actually in the clinical practice because we already know who has CHD. We tested the same with different outcomes. So if different outcomes on the long term, like 2-year outcome, also occupy different spaces, we did not find so far big results in that front. But what we did is we thought maybe we have to move away a little bit from traditional connectomics and dig a little bit deeper into the data and not restrict ourselves to these two dimensions. And this is usually when people start working with machine learning, but let me explain the idea behind that, why we thought it would be great to use a new machine learning approach. So typically when you do connectomics, you need to have an atlas of a certain number of nodes. But on the other hand, the data stems from a single voxel level acquisition, which is almost impossible to analyze because you have very large matrices, very sparse and very noisy matrices. So if you wanted to ignore atlases because you're not sure that your atlas really represents the regions in the developmental stage, or you don't trust your registration, or you have any other reasons not to use an atlas, maybe it's a good idea to look at the original data from which the atlas is a binning of the data anyway. But the problem is that you have huge graphs, you have differently sized graphs because all brains have different number of voxels. But the advantage is if we take the original data from a single individual or groups of individuals, you might not need an atlas and you might have an approach that tries to discover the connectomic structure without the use of standardization. So then we turned to our machine learning collaborators at the ETH, and we started working with graph neuronal networks. So graph neuronal networks are machine learning approaches that take information from the graph itself, a graph representation of data. So while convolutional neuronal network has this filter function, this convolution that goes through the pixels of the image and combines information from that spatial neighborhood, what graph neuronal network do is that they combine information in the graph, so they propagate through the graph. And we thought this might be a way to discover these latent structures that exist in the brain connectome. I'm not going to go into detail. This is now published. Basically, we take an input graph. There's these GNN blocks, which are created by sampling the data by propagating through all the neighborhoods of nodes. And that creates a transformed graphs across individuals. And then there's like a classical MLP, which is basically then able to perform some kind of a task, a prediction or a regression in the end. What we use this for is a toy example, a proof of concept. We thought, are we able to use this voxel level connectome data to discriminate between etiologies? And I know we already know which neonate has which etiology, so it's not much of a useful thing, but we thought this as a proof of concept. So we had congenital heart defect babies, preterms, normal, spina bifida. Also, we combined the DHCP data for this experiment. And we had to look at how this network is able to learn the patterns associated to these diseases within the connectome. And we thought it's interesting to visualize what happens with the data if we embed it into a low-dimensional space with UMAP before doing the whole experiment. So we embedded each connectome just by doing the UMAP. And this is how the different pathologies occupy different spaces. And we basically depict how the different stages of learning separates the etiologies gradually. So at the final layer of this classification task, we realize how these different etiologies are now separated. So this network basically learned what is the difference between these etiologies, or at least we hope that it has done that. So putting into numbers, what we did is an evaluation of the same approach, but using an atlas as a ground truth without the additional information from the voxel level, and also with and without the GNN block, meaning that you can still do a classification on the input graph and feed that as a feature. You flatten out the features and just feed it. And what we see is that the precision of the classification between the etiologies improved even a little bit from without the GNN block on voxel basis, but also atlas versus voxel. So we believe that voxel level information might contain some additional information that can be leveraged. And now we are testing it to find differences between clinical outcomes, for example, 2-year Bailey outcomes. Hopefully, this will be something promising. Now, the last project I wanted to talk about for method development is developmental brain age estimation. So this is a collaboration with a Korean team. Here, the idea was that the biological age and the chronological age are not always in synchrony. So the chronological age during development or during also throughout your life is something that you measure from your birth or from conception. So how old is your brain? But the biological age is something that characterizes accelerated or decelerated aging. This is a very active topic in Alzheimer's research, for example, on the other spectrum. So we thought we try to apply this to development. For this, you need a training data set that is based on a assumed normal trajectory. So what such an approach does, and this is quite popular, so we just published a review on the field in GMRI, is that you take a large sample of normally developing fetuses and infants, you train this dense net machine learning on the MR data to basically zero the brain age gap, assuming that the brain age gap is zero in normal development. And when you apply it to a pathological brain, for example, a preterm born brain imaged at term equivalent age, what you may or may not find is a discrepancy between the two, a brain age gap. So that's the theory. We implemented this approach, and we are trying to extend existing methods to be agnostic to different modalities. So we pooled data from Korea, from China, from Zurich. Now, I talked a lot about methods. Now let's see how we used these methods in our research. What are the latest findings using these techniques? So we are situated in a hospital and have a lot of clinical projects, but we were still interested in normal development. And I have to have a few slides about this. Gregor Kasprian's, one of your favorite topics, I think, is the hemispheric asymmetry. And it was noted quite early by Wittelsson and Paley that the newborn brain has some structural asymmetries. And this was very nicely described in this Cortex paper, that even during gestation, there are some parameters like the right superior temporal sulcus that is asymmetric. So maybe there's like a structural preparation for later functional specialization in the brain. So what we did is a slightly different approach than what was done before. So we had a study where we had longitudinal fetal and neonatal data from the same patients. So we had babies with heart defects and we had the control groups. And Celine from my group was working on an approach that basically quantified the change of asymmetry between two time points. Now, this is pretty challenging, but just an overview that basically we flipped the images, we registered that, and basically we're able to capture the time variance of, or the change of asymmetry from fetal to term equivalent neonatal scan age. So what you see here in this paper, the white blobs are the randomized TFC corrected blobs where asymmetry changed. So fetal and neonatal are the static asymmetries that we found with the Jacobian, and fetal to neonatal is the change of asymmetry. So we were particularly interested if asymmetry is static, meaning that it appears and then just gradually becomes more, or if asymmetry is somehow disproportionately changing during gestation. If you're interested, you can read how we did, but one of the most significant finding was that the temporal pole seemed to change its asymmetry even from late fetal, which is 32nd week, to neonatal, when we were able to image these babies twice. So that's, I think, an interesting addition, and this was possible by the approaches that we were working on. The other one was a PhD thesis from Celine and Hui from the group. We had this congenital heart defect study, and we also was able to use data from the Shanghai Children's. And this has been described before, so you can see this as a validation that during fetal development, heart defect already impacts the brain. What we found is that the gyrification index might actually be a better marker than volumetric differences. And what we also found is that some tissue types, some parts of the brain, such as the white matter you see on the right spot, are probably more sensitive markers for the CHD normal differences in utero than total brain volume. So this is just a glimpse of how we could use brain segmentation approaches, although we did not contribute too much to the field because it's quite well known how CHD affects the brain. Where we could make more contributions is a very special condition called spina bifida. So this is a condition of the spine. There's an open spine and the spinal cord is exposed and damaged. And the brain is affected. So in Zurich, there's a center for fetal surgery. So usually fetuses with spina bifida come to Zurich and they get operated in the womb like this. So they open the uterus, take the fetus out temporarily, close this lesion in the spine with a muscle flap, and then they suture and put it back and the pregnancy continues. And this is quite a unique operation. But the thing is, if this is not done, 50% of these babies are wheelchair bound. And one big international trial showed that prenatal correction improves the motor function compared to postnatal correction because of the damage that's done to the spinal cord early. In Zurich, there has been over 250 fetuses operated with this procedure in the past 10 years. And the very good thing is, at least for my group, is that MR is part of it. So we have a pre-operative MR, post-operative, and a neonatal MR for all of these babies. And since I'm there, we were trying to push this a little bit. So we are doing super resolution reconstruction of all of these babies, DTI and some other sequences. But what we noticed is that, of course, the segmentation methods don't really work for these brains because they're very pathological, as you can see. So during her PhD, Kelly worked on the segmentation approach. She was using some human in the loop annotations of these images to get to a segmentation, a unit base that worked pretty well. And what this data allowed us to do is to calculate the growth trajectories of individual brain structures preoperatively throughout to the neonatal time point, allowing us to, for example, calculate the growth rate of all of the structures that we're interested in. And we did we tested two hypotheses. First of all, the type of the lesion, if it's completely open, what is the height of the spinal lesion, how that correlates with the brain development. We already found interesting things. These are uncorrected values, but I think many of them would survive. We're working on now the paper. So for example, how the cerebellum growth and changes volume is related to the openness or closedness of the lesion. And more importantly, we have a 2-year outcome for all of these babies. Now we're working on 10-year outcomes for those that have. And what we found is that the gross motor functions is related to the ventricular volume. So those who developed a bigger ventriculomegaly had worse motor functions despite the surgery. Cerebellum was also found to be crucial, but also deep gray matter volumes seem to have a link with language. It's closely related to cognition at this stage. Now, another interesting study on the same cohort by Hui published in 24 is we did DTI for all of these neonates, and we were interested how the severity of this condition is linked with thalamocortical development. So is thalamic circuitry or thalamus itself is affected by the conditions, and how is the severity is linked? So what we did is a rather classic approach where we do probabilistic tractography from the thalamus to preset targets in the cortex, and we basically cluster the thalamus based on the strongest connectivity to the frontal lobe, which is red, parietal lobe, which is yellow, and white is temporal lobe, and orange is occipital, I believe, in this visualization. So we calculated the volumes of these connectivity-based parcels in the thalamus and compared the level of the spinal lesion. So L3, L4, L5 means what is the highest level of the spinal defect. L4 means that the baby is probably wheelchair bound, while L5 is not so severe. So that means that the baby can walk. And this is all done at a later, so neonatal stage, while the images are term equivalent. So what we found is a correlation of the parietal thalamic projections and the spinal level, meaning that lower spinal lesion means more connections or larger representation of connections between the thalamus and parietal lobe. And this relates to a better mobility of the patient. So this was done on 50 patients. So far, we hope to be able to do that on all of the patients. Now, moving to the very last part is the brain age gap. We started with already existing hypotheses. For example, this work in Nature Communications and also another one in MILs, where they checked at term equivalent age whether the brain age gap depends on the degree of prematurity. And what they found is that those babies born earlier have a bigger brain age gap measured at the same time. And we were trying to extend this to, first of all, a multicentric database, because we did not really believe that this approach is generalizable. So we wanted to test that. And also we wanted to see how this relates to other conditions such as congenital heart defects. So let me walk you through this picture. We trained our network on the T2 images, but also we were trying to maybe just feed information from cortical development, although we thought the network might pick it up. But nevertheless, we tried what happens if we only feed the cortex into the network. So what we found is basically a reproduction of increasing prematurity leads to a greater brain age gap in the negative direction. So those brains at the same age look younger. But we took the cortical labels, unfortunately, because we did not reproduce this for the T2 images. This is still work in progress. We saw a difference, but actually towards positive. So still, we have to investigate it further. And the more interesting is, I don't think it was done for heart defect brains. So we had a collaboration with Shanghai Children's, over 100 fetuses with CHD and control conditions were involved. So surprisingly, we did not find a brain age gap. And even more surprisingly, a little bit of a positive brain age gap was found for the heart defects. Now, whether this is artifactual or it's truly biological, we still have to investigate. What we also want to do is to have a third center where we could validate if these gaps are real or not. So in fetuses, it might be that it's accelerated aging. And in neonates, we saw a non-significant but negative gap, because we know that these neonates have smaller brain volumes. So maybe the network has picked up this lag behind development in congenital heart defect. So to summarize my talk, I believe computational tools enable quantitative fetal and neonatal MR analysis. We applied generative AI, you remember the stable diffusion, to pathologies. I think this is an interesting way to augment data for downstream applications. I think machine learning helps interpret or use complex connectomic data, especially to embed in a lower dimensional space and understand data differences between groups. And I think all these approaches reveal novel biological insights into brain development, which would eventually help us link pathologies and clinical outcomes to brain development. So thank you for your attention. I'm also grateful for the team. Of course, it's not my hands-on work. We are now grown to be 15 people with 4 PhD graduations, and also grateful for the funding that I had.